Let's switch gears here now and talk about the function of neurons. Neurons are capable of what we call signal transduction. We've mentioned this term before in previous topics. When we talk about signal transduction, we're really talking about a transformation of information. We know that, for example, hormones and things can bind to receptors on the outside of a cell, cause a receptor to change shape, and that shape change can then trigger second and even third messengers inside of a cell to cause something to happen. That's a signal transduction. The information that the hormone has is being transduced or transformed into some kind of an action inside of the cell. What neurons are doing is taking all kinds of information, again, from outside of themselves and transducing or transforming that information into an electrical signal. That electrical signal can then be sent very effectively along long distances in order to transmit that information. Think about when someone steps on your foot. There are neurons in your foot that are capable of receiving the information that is someone stepping on the foot. So the pressure of that, perhaps the pain of that. Neurons on the sensory side are able to detect that pressure and that pain. Now, those signals, the pressure and the pain, have to be transduced into an electrical signal that can then be sent along the length of the axon to the next cell. That sensory information can then be sent all the way to the central nervous system in the form of electrical current. The current is then received by cells in the central nervous system, and some kind of response is made. The beauty of using an electrical signal is that electricity can be transmitted very efficiently and very rapidly. So we need to talk a little bit about this phenomenon of electricity. I know that as students of biology, we sometimes don't enjoy talking about things like electricity because they are concepts that are hard to grasp. We can't touch it, we can't stain it, we can't look at it under a microscope, but it's really important that we understand it, particularly in relationship to a living cell, so that we can understand how the nervous system functions. So, let's talk about membrane potential first. Neurons have what's called membrane potential. Now, when I use that word potential, it's just like the way I would use it when I talk about potential energy. Potential means stored. So neurons have membrane storage. They have energy stored across their membrane in the form of ion gradient. We talked about how ion gradients are used by cells for the short-term storage of energy. Now, all membrane potential is a difference in not just the ions themselves, but a difference in the charge that each ion carries. Remember, some ions have a negative charge, some ions have a positive charge. Ions are going to be distributed across a cell membrane in different concentrations, and that creates ion gradients as well as charge gradients. So we can think of membrane potential as also being electrical potential because charge is involved. Now, when I use that term electric, I'm just talking about something related to the charge on an ion. The potential energy or the stored energy that is created by differences in charge distribution across a membrane is what we're talking about when we talk about electrical potential. So, neurons have this electrical potential, this membrane potential. But what's interesting is all cells have this membrane potential. All cells, not just neurons. It turns out that in all cells, the membrane potential, that electrical potential, that stored energy in the form of charge gradients, comes in at about minus 60 millivolts. So again, the difference in charge creates this stored form of energy of about minus 60 millivolts. Now, the minus just means that across the membrane of a living cell, the cell is slightly more negative on the inside compared to the outside. And just like we can measure electricity in millivolts in our homes, we can measure electric potential in millivolts across the membrane of a cell. Slightly more negative inside, slightly less negative outside the cell. And when we sum it all up, it comes in at about minus 60 millivolts. All living cells have this membrane potential of minus 60 millivolts. All living cells have a disparity or an inequality in the way ions are distributed across the membrane. Different ions are higher inside than outside. Other ions are at a greater concentration outside than they are inside. And all ions carry charge. That's what an ion is. So when you add up all of the positive charge and all of the negative charge inside a cell and compare it to all of the positive and all of the negative outside of a cell, in a living cell, there's going to be more negative charge on the inside than the outside. That's what we're talking about when we talk about membrane potential. Now, there are lots of different ions that we can find inside of a living cell, but when it comes to membrane potential, it turns out there are a couple of very key players. Membrane potential is primarily generated by sodium, potassium, and chloride. So sodium and potassium with positive charge, chloride with negative charge. And there's one more player. And that last player is protein. We know that proteins are very abundant in living cells, and it turns out that proteins have a net negative charge. There are some amino acids that are charged. There are acidic and basic amino acids. And when you add up all of the charge within a protein, proteins have a net negative charge. So they too contribute to this uneven distribution of charge across a membrane. So minus 60 millivolts across the membrane of all living cells. We call this membrane potential the resting membrane potential because for most cells, that membrane potential stays the same. Neurons, though, are different. Neurons can do something with that resting membrane potential that other cells can't. It turns out that neurons and muscle cells are able to put this resting membrane potential into motion along the length of their membrane. And when you put membrane potential into motion, that's what we call electrical current. Take a look at this drawing on this slide. You can see a piece of membrane right here with phospholipid heads and tails in the middle and phospholipid heads over here. The inside of the cell is shown in this peach color. The outside of the cell is shown in this blue color. Now take a look at the ions that are inside and outside of this cell. These are certainly not all of the ions that would be present in a living cell, but you can see many of the most common ones. You can see, for example, sodium, shown in green, that carries a positive charge, chloride, that carries a negative charge. Here's a potassium inside the triangle that carries a positive charge. Inside the cell, you can see some phosphate groups. We talked about how abundant they can be. We can see some proteins depicted by these large red circles. The protein, of course, carries a net negative. Here's some potassium with its positive. So if you start to examine these very common ions and their distribution across the membrane, you start to see where the inequality is. There's a whole lot more sodium outside than there is inside. Lots more positive charge from sodium outside than there is inside. The potassium is the opposite. There's a lot more potassium inside than there is outside with its positive charge. There's more chloride outside than inside. There's more protein inside than outside. So we have unequal distribution of those four particular ions. And again, when you add up all the positives and the negatives, what it produces is a net positive outside of the cell, a net negative inside of the cell. So when we compare the outside to the inside is relatively more negative than the outside is. And if you were to measure this with a voltage meter, it would tell you that it's at about minus 60. Now I say about minus 60 millivolts because this resting membrane potential is going to differ from cell type to cell type, but on average, it's about minus 60 millivolts. So all cells have resting membrane potential, but neurons and muscle cells have the ability to do something with that resting membrane potential. Neurons are capable of what we call conductivity. They are able to use membrane potential to generate electrical current. Now again, those are terms that we're not used to talking about in biology, but we're going to discuss this process, this conductivity, as it relates to neurons and muscles, because it explains to us how the nervous system functions. When a neuron is stimulated, it's capable of generating something called an action potential. And what an action potential is, here's that word potential again. It's a change in the membrane potential, that electrical potential that's associated with the movement of charge, the passage of charge, the movement of an impulse, along the length of the membrane. This is a very unique ability that neurons and muscle cells have. The positive charge, remember we said that is higher on the outside of the membrane than the inside. The positive charge can actually flow along the length of the neuron, particularly along the length of the axon, because that's the direction that the signal will travel. And that positive charge is essentially carrying the message with it. This is possible. This passage of positive charge, this movement of membrane potential. This is possible because of differential opening and closing of the gated ion channels that we talked about when we talked about membrane transport. Neurons can open and close ion channels in a very specific pattern along the length of their membrane, allowing ions to move, allowing them and their charge to move from inside to outside and back again. And by opening and closing those channels in sequence, the neuron can send charge along the length of its membrane. It's a very unusual concept. Remember, cells have control over what comes across their membrane. And remember too that ions, because they have charge associated with them, they can't simply cross a membrane between the individual phospholipids. They need assistance. They cross membranes using facilitated diffusion or active transport. We know that when it comes to facilitated diffusion, it's membrane proteins that are helping ions get across the membrane. And those membrane proteins are in the form of channels. Channels are essentially tunnels through the membrane. Now, they don't stay open all the time, though. They are gated. And just like we talked about the pores in the nuclear envelope having gates, that something has to happen to make the gate open, the same thing is going to happen with facilitated diffusion and ion channels. These ion channels are closed until something happens that opens them up and allows ions to pass through. Ion channels and protein pumps are very specific. In other words, there is a channel for sodium, there is a channel for potassium, there is a channel for chloride, there is a pump that moves protons, there's a pump that moves sodium and potassium. These are specific proteins for movement of specific ions at specific times. The sodium-potassium pump, for example, is sitting in the membrane of almost every cell in the body. It's a very, very abundant pump protein. The membrane of cells is impermeable, relatively impermeable to sodium and potassium as well as other ions because of that charge. We know that ions will be hydrated when they sit in a watery environment like living tissue. And that hydration, that hydrogen bonding that occurs to water molecules is going to essentially increase the size of the ion to the point where it simply cannot fit in between the phospholipids. So, membranes don't let ions just slip across them. These pumps, though, these pumps can help. Now, here's something we haven't talked about yet, and that's this. There is some leakage that occurs this anatomy of the membrane preventing ions from slipping through the phospholipids. It's not perfect. Biological systems are, in fact, never perfect. So there is some leakage. There is some leakage of ions across membranes. That's why having certain pumps distributed widely through the body is important, because if ion gradients need to be maintained for functional uses, then you don't want leakage to occur, because leakage could make those gradients go away. So pumps need to be there to handle leakage. The sodium-potassium pump is going to pump sodium out of the cell while it pumps potassium into the cell. If you remember from a moment ago, we said that under ordinary conditions, there's more sodium outside, more potassium inside. So if we were to allow this potassium to leak out very slowly, or this sodium to leak in very slowly, that gradient, that sodium gradient and that potassium gradient, would go away. So these pumps are critical to maintaining these gradients that we expect to see across the membrane of living cells. The sodium-potassium pump actually pumps 3 sodiums out of the cell for every 2 potassiums it brings into the cell. And just like all protein pumps, it needs energy because it's working to maintain a gradient, to keep a gradient in place. So it's going to consume ATP as it pumps sodium out, potassium in. On this slide, we can see another drawing representing the distribution of ions across a resting cell membrane. Here's the membrane again across the center of the drawing. You can see the phospholipid heads in yellow on either side and the tails tucked in between. The yellow area represents the outside of the cell. The blue area represents the inside of the cell. Now this is a cell at resting state. So the membrane potential, the difference in charge across the membrane from outside to inside, is about minus 60 millivolts. The sodium ions are higher outside than inside, and the sodium is drawn as these small purple circles. There are many more purple circles outside than there are inside. Potassium is the opposite. There are many more yellow circles inside than there are outside. Potassium is shown in yellow. You can measure these charge differences, these ion gradients. And that's what they're showing us in the white boxes. The concentration of potassium, again, the yellow circle, is at 20 millimolar outside the cell and 400 millimolar inside the cell. So there's a gradient here. More potassium inside than outside. It's not just potassium, it's the charge that potassium is carrying that differs between the inside and the outside. If you look at the sodium numbers, there's 150 millimolar sodium outside the cell, 50 millimolar inside the cell. We have another gradient. The sodium, too, carries a charge. Now, in addition to the sodium and potassium, remember we said chloride is important for resting membrane potential, and proteins are important for resting membrane potential. And when you add it all up, when you add up all the positives and the negatives from those 4 things outside of the cell, and compare it to all of the positives and negatives from those 4 things inside the cell, the inside of the cell is going to be relatively more negative. The outside of the cell is going to be relatively more positive, producing this membrane potential. Now, inside this membrane, they've drawn 2 structures for us. They've drawn this, which represents a protein pump, and they've drawn this, which represents an ion channel. This happens to be a potassium channel. Only potassium can pass through this channel. This pump is a sodium-potassium pump. It's going to pump 3 purple circles out, that's the sodium, for every 2 yellow circles it brings in, that's the potassium. And in order to do that work, because it is working against the gradient, it is maintaining the gradient, it needs ATP to do that work. It needs energy from ATP. These pumps are critical for maintaining gradients because, as we just said, this system, the system that keeps ions in place, it's not perfect. And ions can actually leak through these channels. Potassium can leak through its channel. And because channels participate in facilitated diffusion, remember, facilitated diffusion is a spontaneous process. It doesn't involve any energy. So the direction of movement is down the gradient. This channel would move potassium from the side where there's lots of it towards the side where there's not very much of it. It would move down its gradient in order to equalize the concentrations, in order to get rid of the gradient. So yes, this channel is kept closed most of the time so that this gradient doesn't go away, but it's not a perfect closure. Potassium very slowly leaks out, and that's why this pump is critical, because this pump is going to take care of that leakage and help keep the potassium concentration high on the inside and low on the outside. So at rest, across the membrane of any cell, there's going to be different concentrations of ions. There's going to be ion gradients. Leakage occurs through the channels. That's just a feature of biological systems. They are not perfect. The leakage has to be managed, though. The leakage has to be taken care of with the pump proteins, because if it wasn't managed, over time we would lose the differing ion concentrations. We would lose the resting membrane potential. Now I want to introduce some new language here, and these are the terms hyperpolarization and depolarization. These are these are terms related to charge. So during these leaking events through the channel proteins, if we look at the potassium channel, potassium is normally high inside the cell compared to outside the cell. So potassium would leak out through its channel. As potassium leaves the cytoplasm and goes to the outside of the cell, it's not only getting rid of the potassium gradient, but it's also carrying its charge with it. So if potassium leaves the cell, some positive charge is also leaving the cell. And that means that the cell is becoming less positive or more negative. Let me say that again, because that's so important to understanding what's going on with membrane potential. Normally, we expect potassium to be higher inside the cell compared to outside. That's part of what contributes to the minus 60 millivolt resting membrane potential. So potassium can leak through its ion channel to the outside of the cell, down its gradient. As the potassium goes out, it carries its charge with it. So positive charge is leaving the cell and going to the outside. That means that inside the cell, there's less positive. Or you could also say more negative charge. This is a state that we call hyperpolarization. More negative means hyperpolarization. Now think about the sodium. Normally we expect to see sodium ion higher outside the cell than inside. Sodium can leak through its channel though, and it's going to leak down its concentration gradient. So sodium will leak inward. And that's going to take positive charge from the outside of the cell to the inside of the cell. So the cytosol is going to become more positive when sodium leaks in. Or you could say less negative. More positive, less negative. That is a state called depolarization. So in a resting cell, these things are going on. Hyperpolarization, depolarization is going on slowly, but the pumps are managing it. The pumps are managing this so that this doesn't continue to any great degree. Make sense? The cells throughout most of the body are going to manage hyperpolarization and depolarization and don't let it get out of hand. They're going to maintain a membrane potential of minus 60 millivolts. But here's the key point. Neurons are going to take advantage of this. Neurons are going to use hyperpolarization and depolarization to their advantage so that they can put charge gradients into motion along the length of their membranes, creating electrical current that can be used for signaling purposes. So, if a cell became suddenly very permeable to sodium, sodium flowing through its channel into the cell, that could be looked at as a purposeful depolarization. If the cell were to suddenly become very permeable to potassium, flowing out through its channel, that could be considered a purposeful hyperpolarization. This is exactly what neurons do. Neurons are going to purposefully open sodium channels and allow sodium to rush into the cell under certain circumstances, causing a depolarization. And simultaneously, although at slightly shifted times, they're going to suddenly open up potassium channels and let potassium rush out, causing a hyperpolarization. And by carefully coordinating these two things, the exact timing that the sodium channels open and close, the exact timing that the potassium channels open and close, the neuron is going to purposefully create depolarization and hyperpolarization states that will allow charge to flow down the length of its membrane. This is how neurons create electrical current.